Is Dr. Meg Macy. She did her undergraduate work at Middlebury College in Vermont and received her MD from Case Western Reserve University School of Medicine. She completed her residency and fellowship at the University of Colorado Children's Hospital. Her clinical and research interests are in new therapies for hard-to-treat pediatric tumors, including solid tumors and brain tumors. She has a particular interest in relapsed and refractory neuroblastoma. She is the author and co-author of numerous articles on new drug therapies, an investigator on several neuroblastoma clinical trials, including those through the NANT. Dr. Macy will be talking about the drug development process and how they get new therapies to our children. I'd like to welcome Dr. Meg Macy.
Thanks, Pat. I want to thank you for the opportunity to be here today to speak on this. This is a little bit of a new talk for me. I do pediatric drug development on a daily basis, but trying to put it into a talk about how drugs go from the lab into clinic and all of the problems and challenges was actually a new experience for me. So bear with me if I trip over my words a little. So this is the overview of how cancer drug development happens. And this is actually a slide that I give my fellows the first month that they're fellows to teach them. And it's very long, but basically it starts at the very beginning with cancer physiology, so understanding what's going on in the tumors, and then trying to find the targets, identifying through various screening techniques, which, to be honest, even though I have worked in the lab, sometimes those are beyond my skill set. Then trying to figure out how to make those drugs work best by chemistry, and then do a bunch of pharmacology studies, which you need your pharmacologist for, because that's definitely not in my skill set. Then you need a company to make the drug. Then you had to figure out, is the drug safe, how to even give it to patients. Then it goes to phase 1, phase 2, phase 3, 4 clinical trials. Then it gets approved. Then it gets into general practice. Then we figure out standard of care. So it is a very long and windy road, if you will, to go from somebody's idea in the lab and forward. So one of the things that I harp on a lot in what I do is why it's important for pediatric patients to be considered in drug development and to be considered early. And when I started many years ago now, it was rare for pediatrics to even be considered until the drug was at least in phase 3 adult trials. We have moved that earlier. There's been a lot of work being done in pediatric oncology where companies are thinking about pediatric drug development earlier. And I'll talk a little bit about the rules and regulations that have helped make that happen. And why we're better now, but it's still a long process. But kids have done better with pediatric cancer than adults have ever done. And part of it is because we do large trials. You heard Dr. Mathey talk about COG, you'll hear Roz talk about NANT. We do better in treating pediatric cancer because we treat them all the same, we treat them consistently, and we learn from when we treat. Every patient gives us information. We learn from every patient we can, and as a result, we have made huge strides. And I mentioned COG and NANT, but there are many consortium that do this, and really, we can't do this alone. It is not like an adult center where they can treat 500 breast cancers and come up with their own studies. We just don't see that many patients, thank goodness, but we have to partner together. And it helps us learn about the diseases, helps us learn about the toxicity, helps us learn about the long-term effects, and really approaches the disease in the same standard question. So we can say, we know X is better than Y. Otherwise, how do we know this is better than that if we don't compare them or at least look at standard things? We have to standardize how we approach it. Now, I stand up here saying that. I also say every patient is different and we have to consider what's best for that patient and how that fits in these trials, but there is a real benefit to standardizing how we approach. So kids are not little adults, and these are my niece and nephew. I pirated this from my own thing, but you can see my niece is wearing my shoes. She was about 4 at the time, and my nephew somehow forgot his pants. But kids are not little adults, so you cannot take what works in adult world, either dosing-wise or how their body metabolizes, and put it into kids. You can't. We all know that. It's all pretty logical, although the Doubleday Junior pills, and I don't know, this is really old dad, but they didn't seem to think that. Their bodies change between infancy and adolescence. How much body water a baby has versus how much body water your 15-year-old son has is very different. That means where the drug goes is very different, how their body metabolizes is very different, their kidneys work very different. You know, if you've all had infants. You can't give them just water. That's bad for them. But you tell your 15-year-old to go drink 25 gallons of water and they'll be fine. Their kidneys are different. So the other thing that's complicated about pediatric drug development is kids are growing. Their cells are changing and they're making new cells over and over again. We're kind of stuck. Yeah, we make new skin cells, we make some new GI lining cells, but we're not getting bigger bone cells. We're not doing all that. So the effect of these drugs on the growing cells is much more important when you're looking at pediatrics, and much more important to consider when you're developing a drug. And the other thing is kids are different sizes. You don't want to treat someone this size and someone this size with the same drug. They're going to have different levels, different toxicities. And many kids cannot swallow tablets. That is a huge problem with many drugs, because they come in one size fits all and it's a tablet. And half the time, I swear to God, they're the biggest tablets you can find, and the stickiest or worst tasting tablets. Decadron is like the disgusting tab from. But anyway, that is a big challenge. I know you guys are probably all really good at hiding things in chocolate syrup or cherry syrup or something. So I've now given you the argument why I think we should test them in peds, but where does it start? So it starts in the lab. We call this the preclinical. And forgive me if you guys all know this, but I thought I'd start with this. And it used to be that back in the old days, back in the 50s, 60s, whatever, 70s, they were all looking for drugs that killed rapidly dividing cells, right? That's classic chemotherapy. Classic chemotherapy is what can kill cells that grow fast and die fast. So that's historically what there was. And so they'd put them in the cells, they see the cells would die, and they'd say, okay, well, this is a good drug, and it seems to affect faster growing cells than others. But with an increased discovery of the Genome Project, understanding the genes that people have in their bodies, understanding the genes that are driving cancer, people have really moved away from what we call the traditional cytotoxic or cell killings chemotherapy, and are much more interested in targeting molecules that either block pro-growth, so things that turn on growth in those cancer cells, block molecules that help the tumor resist chemotherapy, so either spit the chemotherapy out of the cell or not be sensitive to it or something along those lines, to turn on pro-death signaling. So some of the cells just don't have the mechanism to die. They don't ever have you're done growing. So some of the molecules can target that. And then the other one, which has been really big in the news for past decade or so, is stimulate the immune system to better recognize and kill the tumor. How do we teach your immune system that the tumor is foreign? Right now, your tumor is pretty good at figuring out how to hide them from the immune system. How do we teach them? And they do them initially what they call in vitro, which is usually in a dish or in a bottle or something. You're just doing it on the cancer cells themselves. And I like to joke that we've been curing cancer in the lab for years, but it is much easier to kill a neuroblastoma cell in a petri dish than it is to kill a neuroblastoma in a mouse, and then it is to kill a neuroblastoma in a patient. And part of that is because there's a lot of other factors involved. So when you start in the dish, you're really just looking at the cells. And then you put them into the animal and you look at a little bit how it interacts. And then you put them into the human, which is much more complicated. And so we can cure neuroblastoma in mice. The challenge is how do we take that forward and cure them in the humans? So as I said, we go from in vivo to in vitro to in vivo, and that's really looking at, is it even tolerated? Did the mice tolerate it? Is it having the effect we want? But usually it's not a great model. We're injecting the neuroblastoma. Mice don't typically get neuroblastoma on their own unless we do something to their genes. So it's kind of, they're all a little bit of a fake model. So you use the best you have, because we're not going to be testing scary drugs on people until we know they're at least safe. But that's kind of the concept, is you do the best you can until you can get into the lab. And as I said, we've been curing mice for years, but this mouse says, I'm going home. They cured me using a numerical drug. I'm afraid it'll be years before it's approved for humans. And unfortunately, this is really quite true. So what happens when the drug is ready for prime time, ready to go into humans? So first one is the phase 1 trial. And so that's usually a first-in-human trial. There is something called a phase 0, where they'll take med students who need money, and they'll give them some money and give them some drug and see what happens. But generally, you laugh, there's lots of them that do it. But so in a phase 1 trial, the goal of the phase 1 trial, and I tell this to families who come to see me, is to figure out what is the safe dose and what is the correct dose to give to patients. It's not, does this drug work? It is, how do we give it safely and for your patient, and to learn more about how the body metabolizes it. So usually it's restricted to patients who don't have any other options. We're not going to offer this, and we shouldn't, and it's ethically wrong to offer this to someone if they have something that we know is safe and effective, because that is what we should be doing. But this is more when we're kind of out of options and we're looking at things. This might work, this might not. We just don't have any of that data. We're really trying to figure out what's the right dose and how do we give it. They increase dose in different steps in different patient groups. You heard someone talk about a 3+3 design. Basically, you enroll 1 to 6 patients on a dose level, look at the toxicity, look at how well it's tolerated, and then increase that dose level again. Some studies along the way will look at the, is the target, if you can figure out what the target is the target being hit properly? And so they won't go above because they know that they're completing that target. Other ones are really how well we push it till it's maximally tolerated, till you see toxicity that you don't think is acceptable. The toxicities are, you know, usually they grade them on a grade from 0, which is no toxicity, to grade 5, which is death. We don't ever go to grade 5, please. But a grade 3 toxicity is usually something that is going to cause the patient to have some change in their lifestyle. So they're going to, you know, let's say it's a kid, they're having enough pain, they're not going to go out and play, they're adjusting their care because they can't do that. That's a toxicity that we would call a grade 3. There's different rules and regulations and how you change it, but that's why if you're ever on a phase 1 or phase 2 trial, and if you're at my institution, we will quiz you. When did it happen? What did it start? What did it do? How bad was it? Where did you, because we want to know that to be able to make sure we're giving that drug safely and we're capturing all the toxicities. So you get to see your phase 1 doctors a lot if you do a phase 1 study. It's small numbers of patients, somewhere between 12 to about 40 patients. We cannot use any meaningful statistics out of this. We can't come out and say it's 40% response rate. Sometimes we fudge a little bit, but that's not what the study is designed for. So we'll take what they call an early signal. You'll hear people say, well, there was an early signal that this had a good response. And that's just, we look at, did we see responses? Now, the drug company people on the, will tell me that they want an early signal because that's going to make it go forward, but that's really not the intention of the study. But it is always a good benefit. So we always mandate that you have to be able to follow the disease. So usually you have to have disease going in. You don't go into phase 1 studies without disease. And then we define appropriate doses and toxicities, as I said. So phase 2 trials, these are typically for first or second relapse. They're larger trials. They're somewhere between 40 to 100 patients, depending. And this is where we know the dose of the drug. We know the general side effects of the drug, as best we can tell from the 40 patients we've treated or whatever. And now we're trying to see, does it work in X tumor or an X tumor with X mutation? So there are much more specific guidances about who's eligible. You have to have only had so many relapses. You have to have had this mutation. You have to have this type of tumor. And this is really the one that's designing to see, do we see a difference? Do we see a response in this? We're not comparing to the standard. We're comparing to whatever we decide to compare to or by itself. But that's the goal of a phase 2. And there's very well-defined statistical rules, and I'm not going to bore you with all because I don't know all the statistics, but there's usually a halfway stopping point where we look and see, is it not meeting at least the minimum efficacy response, or is it way exceeding it? Because if it's way exceeding it, we're going to stop and say this works, and if it's not exceeding it at all, we're going to stop because it's not worth it. And I believe you asked about the 1821. There is a built-in interim analysis at that to see, was arm A better than arm B? Was there a great difference? So that's when it was built into the study intentionally. So we do that for these studies. And very positive results can lead through breakthrough designations. So you will have seen, if you follow pediatric drug development, that there have been a bunch of drugs recently who have been approved sort of early breakthrough designation. It's because their phase 2 had really promising results. The companies were able to go to the FDA, show that, and the FDA gives them breakthrough designation, which says, you're approved, but you have to now still continue to prove this. And breakthrough designation does not mean it ultimately works. There have been many drugs, especially in the adult world, where they get breakthrough designation and then it gets pulled back later on when they do the larger studies. But it at least allows that drug to be available to patients earlier while they're figuring that out. So phase 3 trials, these are the larger trials. These are those big COG trials that Kate was talking about, that Greg was talking about. These are those big trials, hundreds of patients. Really, they're for upfront, and they're comparing the standard of care, what we know works best, with something that may be slightly better or something that may have less toxicity. So you can kind of compare either ways. And that's really to figure out what's the new best standard of care for that. They're often randomized. They can be blinded. It's really hard to do blinded trial in pediatrics because it's just complicated. But you can do blinded, or you can do what they call open label, which means 1531. You know if you got randomized to MIBG. I don't know how you would blind to MIBG. That would be really unfair to the kids who didn't get MIBG and got put in the room. So, I mean, that's a bit of an extreme example, but the point is you can blind or not. And that really is determining what standard of care should change. And then if it meets the endpoint, this is usually what meets the registration approval with the FDA and then the EMA, which is the European Medical Agency, which I will use in a little bit. And then there's phase 4, which are post-marketing. So after it's been approved, usually the FDA mandates certain tests to figure out long-term side effects, other things that they may not see until they treat thousands of patients. And it's really small, specific data. They're doing that with the COVID vaccines now, so it's right FDA approved. They're still collecting data on toxicity. This has happened with some of those COX-2 inhibitors, those that they discovered after the fact that patients had more cardiac issues, and then they put a big black warning on it. It was from the post-labeling, post-marketing phase. So as I said, pediatric development is a challenge. Access to agents has always been hard. It's really hard for a company to want to develop in pediatrics if they don't know that drug is going to go forward in adults, because fortunately, pediatric cancer is rare. Unfortunately, that means financially it doesn't have the cachet that a prostate cancer or breast cancer. So they need to make sure that drug's going to work, or they need to be committed, as some of the companies who are here actually are, to pediatric drug development and the cost and how that works. So, but the big companies, generally, they look at the bottom line, and if you're going to treat 200 patients a year, that's not like treating 20,000 patients. So getting those drugs have been hard. They don't want to start in pediatrics. And the other thing about pediatrics is no one wants to have a pediatric bad outcome. No one wants a kid, you know, that child who died of the vaccine, not that, was gene therapy trial many years ago. It was back when I was in college. And it happened at Duke, and it was like all over the news, and it was like the world's worst thing because a child had passed away. So companies are very gun shy about having a pediatric bad incident. So if they tried a drug and there was bad outcomes, it's concerning to them. And so that's also part of their development reasons. It's not malicious, it's just they don't want to look really bad and have something bad happen. So for years, our trials have used repurposed FDA-approved drugs. DFMO, that drug is approved for sleeping sickness. It is not approved for cancer. It was not developed for cancer. It was approved for sleeping sickness. We have repurposed it into a drug that's being looked at in neuroblastoma. We have been very smart in pediatrics, how to flip things, how to look for drugs that are approved in other indications and flip them into pediatrics. Prior to 2017, only 4 targeted therapies had received pediatric indications on their label, meaning at that point, we were well into development. We knew a lot of drugs. Lung cancer had stuff, colon cancer had stuff. Only 4 of those drugs had pediatric indication, meaning that they hadn't done the work to actually have the pediatric indication. Didn't mean we weren't using it, because we use a lot of drugs off label, but only 4. But since 2017, so just to make you feel a little better, we've had 12 that have pediatric indications. And these trials all had ped development in mind early. So either they knew there was a pediatric target in it and they'd well defined that ahead of time, so allowed kids down to 12 come on the study, or quickly opened the phase 1 and then right after opened the phase 2, the phase 1 for peds. And the majority of these had amazing response rates. So they really enriched for the kids they thought were going to respond. They weren't just throwing it at every kid that walked in who had cancer. They were really thinking about how do we design this rationally and smartly. So this is a timeline that I took from Andy Pearson's work, and it was just basically showing how drug development happens. And I am trying to point with nothing. Oh, there is a pointer here. So early drug discovery, new targets, all this we talked about is the lab. So then it goes into the adult trials, and you see usually now after early phase, it's starting to go to early phase pediatrics. When I started, it was really into late phase before it even got in. This is a 3 to 6 year timeline. This is an 8 to 10 year from the start of here to getting over, because we have so many fewer patients. We can't enroll thousands of patients at a time. Then you have the marketing authorization in children, then you get access, then you get post-marketing surveillance. So it takes a long time to get into the real world. This has been shortened by various things we've done. It used to be much longer, but it still takes time because we have to be able to do these properly and well with the appropriate number of patients. Now, can we be smarter about how we do it? Can we use the brilliant statisticians? Yes, and we should try to do better. I'm not saying I'm happy with this number, and I would say the 8 to 10 is maybe down to 6 to 7 for some indications, again, with a really clear role. But so the FDA and the EMA have recognized this as a problem, partly because of families like you guys lobbying their Congress people that this is ridiculous and unacceptable. So they have what they call what I call the carrot and sticks. They have incentives and then they have regulations. And so back in 2002, there was this Best Pharmaceutical Act that if you had a drug and you looked at it in kids, you got a 6-month patent exclusivity extension. 6 months, you say, oh, no big deal. Oh no, that's millions and millions of dollars if it's already approved for adults. So that's actually a huge patent extension. It's a huge deal. And I apologize to the drug people if I'm giving the numbers wrong, but in my head, this is how I think about it. And then in 2012, they changed it to even more of a carrot, but basically, if you obtain FDA approval for a pediatric indication, you get what they call a voucher. And this voucher can either be used for another drug to get FDA to look at it faster, or you can sell it on the market and give it to another company so they can use it to their drug, and then you get capital to raise money for another to use for whatever. So this was used by UT, I believe, for the dinutuximab. I think they got a voucher for that, and it was used. Don't ask me how they used it. You'll have to ask the UT people for dinutuximab. But this is a big thing as well. These don't sound very big for those of us who don't do business, but it actually is both big deals. So then there's the stick, right? So we're going to make you do this. So in 2005, they mandated that pediatric studies have to be in process at the time they went for FDA. So that moved it a little earlier. They weren't waiting until they had approval and said, okay, now we'll look at peds. They now moved it earlier. And then in 2006, the European group, and this has actually caused a lot of problems, mandated what they call a PIP, or a pediatric investigational plan, which is really the company, when they want to develop X drug for colon cancer, let's just say, if it has a target or something that they're ever going to think about using in pediatrics, they have to go to the European, not only that we're going to do it in these trials in adults with colon cancer, we're also going to develop it in peds, and this is our plan, and we're going to develop this trial this way, this trial this way, this trial this way. That's really hard to do because you don't know what your responses are going to be. You don't know what's coming. I don't have a magic 8 ball to be able to tell you what's going on. So, but the EMA mandated this. And then it was before anything happened, whereas the FDA wanted it later on in the timeline. So now you have Europe directing the trials early, FDA directing the trials late, and things were just getting mishmashed. So that has actually been a challenge. But if you got your PIP completed, again, you got a patent extension. So they allowed waivers for orphan diseases or for diseases that were going to be rare in kids. So colon cancer is rare in kids. They could just say, I'm getting a waiver, I don't need it. So many of them didn't have to require a PIP. So they were saying, well, we're never going to develop in kids. Well, now they just shut the door on pediatric drug development. So that was not quite what they wanted. So what happened was the EMA published actually a waiver list of who could get waivers, what truly were drugs or areas or targets that didn't need a pediatric indication. And then the Race for Children's Act in 2017, which was a huge act in the US, basically required an earlier submission. So you applied earlier than you used to. And you had to do it if there was a target that was relevant to growth and progression of pediatric cancer. And they got together a group of pediatric oncologists, the National Cancer Institute, the FDA, and they came up with a list of over 200 targets that we as pediatric oncologists think are relevant. So no longer were we letting the adult people say what was relevant in peds. The peds people were saying was relevant in peds. And that has really changed because these are now not getting waivers. These are actually having to have investigational plans. Oops, did I go backwards? Oh. So one of the other challenges, as I mentioned, is that peds is a small number of patients in general. So now we're being told that all these companies want to develop these drugs in kids. And there's, the GD2 antibody is a great example. We have two antibodies which are approved for slightly different indications. What if someone else wants to develop a third one? How are we going to put that in there? How are we going to test that? How are you going to test the 75 target drugs that target one specific mutation? We don't have enough kids. So we have tons of drugs now, and we don't have enough patients to be able to meet the requirements that the companies are being able to do. So this is, again, where we have to think smart and think clearer about how we do this. And so how do we maximize our ability to pick the right drug while minimizing exposure for those kids to drugs that aren't going to work? We don't want to be I don't want to be just using, kids are not guinea pigs. Kids were trying to do this to figure out an answer. I want to maximize the chance that if I give a kid a drug on a study, that's going to work. I don't want to just be doing it to see how many different things I can do to meet a company's investigational plan. So what have we done? So one thing is now patients 12 and up are eligible on a lot of adult trials. So there are trials that allow down to 12, which means I partner with my adult colleagues for some of those trials, so we'll have the 12 to 18-year-olds open in our clinic, in our institution. Other ones will send them to adults. But that 12 and up, they're pretty much built like adults. I mean, their brains aren't built like adults at that point, but they're size-wise, their body metabolism, so they can safely be tested. We're at least getting information in those teenage kids. We have a really increased presence in pediatric oncology in the drug development world. I can't think of any company where there's not somebody who's got a pediatric oncology training that is now in there working on developing how do they include peds. 12 years ago, nobody cared about peds. I mean, I don't mean to say that flippantly, but it's true. It was an afterthought. Now, every single group have recruited high quality physicians to be on their team to really drive the pediatric plan. That is really important because it means that the companies are thinking about pediatrics early. How are we going to build them in? How does this mean? How do we answer these questions? And then there's these great groups. There's this Accelerate platform. I don't know if any of you have heard of it. I think some of you guys have been on some of the calls. So it started out of actually Europe, but it is a group of industry, regulatories. So FDA, Greg Rieman's there from the FDA. Don't ask me what the guy's name is from the EMA is there. Parent and patient advocates are a large part of it. There are several of them on there. Pediatric oncologists, of course, basic scientists. And they sit together and they have topics. I think we're doing one on PI3 kinases where we say, this is the target. Where does it fit into the pediatric world? How should we develop it? How should we partner with industry to develop it so we're not doing 12 studies for the same drug or similar drugs? What do we need to know to be able to move us forward? It has really been helpful, and they have, I think, a requirement within 6 months to have a paper out from every single one of these meetings saying what their goals are, so that it's in the press, it's in the journals, saying this is what we think should happen from a pediatric standpoint. And one thing you hear routinely is pediatric-specific testing in the lab is critical. We don't know until we test in those areas, and so that is really, really crucial, and that's one part that has really been stepped up, is really looking at the pediatric testing before it even gets into patient, because we have to maximize those. So this is a thing from a paper that I was part of that really talks about the challenges of pediatric drug development. I don't think any of you want to read it. It's very long and boring, but this just shows you that it is a hub and spoke model, if you will. It takes roles from academia, regulatory, patients, and pharma. And I can't speak enough about the patients because you guys are the advocates. You guys are the ones who go and tell people, we need this, we need the funding, we need you guys to focus on this. It is a crucial part of this, and it is just as big a circle as the other ones. And so I think that's the important part of this. So what about neuroblastoma? Okay, Meg, you've just droned on and on about really boring things, but what's happening with drug development in neuroblastoma? Well, that could be a talk and a half or more on all of the new agents and where they're going and what they're doing. And you'll hear some when you hear about NANT. But I think the anti-GD2 therapy, everyone talks about CAR T cells, but let's be honest, anti-GD2 therapy was the first successful immunotherapy given in pediatrics. That is a huge deal. I tell my fellows, you don't realize that neuroblastoma, forget CAR Ts, neuroblastoma was the cutting edge that showed that immunotherapy worked in pediatric cancers. So it has 2 drugs, but have FDA indications. It is a great example of successful pediatric drug development, successful partnership with industry to really move this forward. So ALK inhibitors, another success. So you heard about the study yesterday of 1531, which included crizotinib, which was a early ALK inhibitor, and now we've moved on to lorlatinib. We did the phase 2 studies. We were able to quickly, you'll hear from Araz that the NANT study is still ongoing, and we were able to throw it into the high-risk trial because we thought about it and it was rationally designed. So 2 big successes. MIBG, okay, it's been around since the dawn of time, but now it's in phase 3 trials. We're really moving forward on that. And then there's so many other agents in phase 2 trials, and some of them have specific neuroblastoma cohorts. My interest is relapsed and refractory tumors. I'm always thinking, what's the next agent we're going to combine? What are we going to do? How are we going to move us forward and really see if it works? Oops, why is it? I keep going backwards, that's why. Buttons. So this is a slide from a talk I gave that I was given the topic of relapsed and refractory neuroblastoma therapies. It's a really busy slide. I don't expect you to know anything about this other than to see that there are all of these things are different targets that we can hit, and we have shown, at least in the lab, has some effect on neuroblastoma growth. So there's tons out there. How do we prioritize which is most important and how we're going to combine them is going to be the challenge. So one of the really good things about neuroblastoma is it's a highly collaborative parent community, and there's a highly collaborative provider community. We work really closely with our colleagues in Europe, our colleagues in Japan. We have meetings every 2 years. It is a very, very active group. And there actually was a neuroblastoma new drug development strategy initiative where ITCC, which is the European Early Phase Clinical Trial Program, CIAPIN, which is the European Neuroblastoma Group, and COG, and they identified 22 out of 40 targets that they took, like, of the 40, which 22 were the highest priority. They published this, and then these are the things that they're holding people's feet to the fire for. These are the things we want to see developed. One of them is this, this I apparently cannot find buttons, telomerase maintenance mechanisms. No one's really talked about that today or yesterday, but telomerases basically are what make your chromosomes shorter. When they get short enough, your cell says stop growing. Please excuse my very basic explanation of this. Neuroblastoma, many of the cells have mechanisms to make those telomeres not shrink. They stay up. They maintain them long, which maintains that cell growing. It's becoming more and more clear that it's important in neuroblastoma, especially the aggressive neuroblastomas. So the NDDS said, you need to figure this out. You need to figure this out. There is a new trial in COG looking at ATR, which has a role in telomerase maintenance mechanisms, some of them already in phase 1 trials. You can see that things are happening. We're moving things forward because we're identifying them. MICN. MICN's really hard to target. We don't have a good drug that goes in and binds MICN and turns it off. We don't. We've all done sort of, back end things where they're now saying, we want to be able to directly target this. You need to spend your money, you need to spend your effort to target MICN because that's really crucial, especially for the little kids. And then how do we make GD2 and chemotherapy combinations better? Daniel talked about this yesterday. How do we add more chemo to it? Is it a certain chemo? Do we add new targeted agents to say, well, we'll target this mutation plus this, or do we make the immune system better? All of those were their 3 priorities, and I will tell you, all of those have studies currently out there in the community looking at these things. So there is direct involvement of the neuroblastoma community to drive this forward quickly. So in conclusion, it's to really develop drugs well, you need solid science and lab-based data, and I can't stress that enough. I don't work in the lab anymore for a reason. Test tubes don't talk back to me. They don't play with me. They don't laugh at my bad jokes. But they are crucial. Scientists are crucial. We need to have good data. You need to have well-designed studies. I can't harp on this enough. If you have a study and it looks like it's positive, but it's not been designed well and it's not been designed to answer the question, it won't get FDA approved, and it doesn't give us the things we need to leverage that to move forward. So we need great statisticians. If any of your children want to be statisticians, there's a great need for high quality clinical trial statisticians. They are the limiting factor in a lot of these. And then teamwork. I like to say there's no egos in pediatric oncology, and it's probably not entirely true, but we work together. I said that we talk to each other. I do. I talk to my colleagues a lot when I have a question or how does things work. We work together. We sit. One of the worst things of COVID is we haven't been able to have our lunches where we sit and babble about something and what about this and how about that and how do you bounce that off. That is crucial to how neuroblastoma has been so successful, and that partnership is important. So it's a partnership with industry and regulatories. Isn't the us versus them. It's how do we move this bar together? How do we move this needle? And then willing patients and families. If families aren't willing to be part of this work, we can't do any trials. So they are the keystone. They are the importance. They're why we do what we do, but they're also really a crucial partner. And that's why I think this is such a great meeting, because it really brings that partnership forward. And we've made great strides, but the only way we're going to do this is to ensure that these continue to move forward and that we hold everyone's feet to the fire, my feet included, to move this forward. Because I think as we do that, we're going to see strides. We've seen some. I want to see it go forward. So in summary, I just want to thank all of the patients and families that have participated in trials and research. Even if it's biology trials, everything it counts. And then I want to thank everyone who has participated from a consortia level or investigators, or I should have also thanked the industry, who've contributed to research in neuroblastoma. It takes a village, or beyond a village, and that's really how we make our strides and how I anticipate we're going to go forward from then on. And that's all. Sorry, that was a lot.
Thank you. Does anyone have questions? Yep, please.
Good morning again, Megan Nelson from Children's Hospital Los Angeles. Thank you, Dr. Macy, for that extremely, informative and enlightening presentation, and also letting us know what we can do as parents to get involved, whether that's lobbying our our congressional representatives. I'm interested to know more about the accelerated platform and how parents can be involved in that as well. 2 questions. You were speaking about the phase 2 trials. I wanted to know if there's any, I guess, magic number for that breakthrough designated approval. Does one say, okay, if we have a 80% response rate, that's designated for breakthrough approval? And second question is, does neuroblastoma have any drugs that would be considered, one of those breakthrough, approval drugs?
So trying to think if neuroblastoma has specifically. I think dinutuximab was in its, I don't know if it got breakthrough designation initially, and I can't speak about NACS, so I'll ask the YMABS folks who are here. Was that breakthrough? It was. So naxitamab was a breakthrough. So what response rate requires a breakthrough designation? That is a question that is, it depends upon how low the bar is. So if you have, and I'm going to use so I used to do brain tumors before I went into neuroblastoma. They both have the word neuro in them. They're very different, but there's a brain tumor that is uniformly fatal, right? So if you had a 30% response rate in that would be a huge change, right? So in neuroblastoma, first relapse is usually about 30%, 40%. We were really excited when we had the dinutuximab irinotecan and it hit almost 50. That was really exciting for us for a response rate. I think in neuroblastoma, if you're hitting 60, 70, 80%, that's something, again, it's a lot of conversations with the FDA. Thank God I don't have to do. The industry partners have to do. But is this enough to be considered for breakthrough? And, again, if it's an in-need population, it's an orphan disease. All pediatric cancers are orphan diseases, just so you all know that, because our numbers are so low. They are more willing to have a lower number, But many of those 17 that got approved were hitting numbers in the 70 to 80% response rates. Again, variety of tumors, things like that, but they were hitting high percentages.
Anyone else? Kimberly?
Good morning.
Nope. Don't hurt yourself, please.
Good morning. Kimberly Turner, Columbus, Georgia. Anytime you talk about something that is experimental and putting kids in the same sentence, to parents, it sends up red flags. So how do you know which kid needs, to be experimented on? I hate to use that word, but it is what it is. It's being experimented on. You don't know a lot about this drug. And as parents, we're scared. I know you need our participation and you need our kid, but in our mind, we just want you to fix them. But after a relapse, because my daughter has relapsed and she's 14, so it is different from a baby who has relapsed. So how would you even know that my kid has relapsed for the second time and they need to have a different treatment? Do the hospital alert you and say, hey, this kid is a relapse, they may be an option for you to try this? And what are you going to do to ensure me that this drug isn't going to speed up the process of my child dying or being hurt more than they already are, versus me saying, okay, enough is enough, it's time to call in hospice or whatever? What are you gonna do to ensure me that this may be helpful and prolong my kid's life instead of having long-term side effects?
Those are all fabulous questions, and I didn't touch on that side for that piece, but let me touch on a couple of things. So first thing, I don't know who's relapsed unless somebody reaches out to me. So either a family reaches out to me through whatever, and I think, we talked about second opinions. Families reach out for second opinions. What do you have for my child? They've relapsed. Or a physician will say, hey, what do you have available for a patient with relapse disease? So that's how I find out about them. I don't go searching. I would be a HIPAA violation and it would be really unethical of me. We have a policy at my hospital. I don't treat upfront neuroblastoma patients for a reason. The primary oncologist has to refer them to me, meaning they have to have a conversation with their patient saying, do you want to learn about experimental drugs? If they say, no, I don't want to do that experimental thing, that's the end. They don't even come to me, because I don't want the doctor-patient relationship that hopefully you've created with your doctor to be clouding your judgment. I have had families say to me, "You're the doctor, you make the decision." That's not allowed. It's unethical, and I work really hard to not do that. So if you come, and I can only speak to what I do, but I think most of my colleagues would do the same. When you come to me, I say, these are your options. You could do X standard thing. We could mix some chemotherapy up, try this, that. These are the clinical trials. I go through what I just went through with you guys on what's a phase 2 versus a phase 1, what are the goals, and then I explain how I think about it. So typically, I think about a phase 2 is less experimental than a phase 1. So if I have a phase 2 that makes sense for your child, that would be what I would think generally they would want, with the exception if there's some crazy indication to think that this phase 1 trial might be perfect for your child, we would have that conversation. But ultimately, then I sit down with you. It's a couple-hour meeting where we go through the risks, the things I know and the things I don't know. I don't know if it's going to accelerate your child's progression. I don't know if it's going to give them long-term side effects. I can tell you what I know based on adult data, what I know based on mouse or dog data, and what I know about similar drugs. But then ultimately, it is a conversation with the parent and an older child. If they're 15, they have to give their assent. Anyone over 7 in our institution has to agree to be on a study. You know, a 7-year-old conversation is going to be very different than a 15-year-old conversation. Then it's a decision on the family's part. It is no skin off my back if you choose that it's not right for you, because the opportunity to have these trials is available to you, but it is not a mandatory, and it should never be a mandatory thing. It should not be, I'm going to treat you, we're going to treat your child no matter what you choose. My goal is to give you the opportunities to feel like you've had that option. If you decide no, great, but will it make you sleep better at night that you know that you were given the option and you chose this because this was better for your child? And then the primary oncologists talk with the families as well if they have questions, because that's their role. Their role is to support you through their whole care. My role is to offer these studies, discuss with you the pros and cons, support you in whatever decision, but their role is to really help you figure out what's best for you. It is a collaborative, and it should be a collaborative interaction. It should not be anyone saying you have to go on this study. I just think it's my job to be able to offer the opportunities for families. And if that's what they want to turn over every stone, I want to be able to offer that opportunity to be able to move science forward.
Okay, anyone else? Dr. Kushner?
Really enjoyed your presentation. I think it's worthwhile pointing out, though, in terms of a phase 1 trial, that it starts off with extremely low doses of treatment of the agent. So it's really unlikely that at extremely low doses, based on laboratory studies, that you're going to have serious side effects. And the doses are only increased when you're sure there are no dangerous side effects. And in terms of phase 2 trials, the side effects are already well defined. I think that's what I wanted to say.
Yeah, and I would say the one thing about pediatrics, if we don't do it ahead of time, is the adults start at like a dose where you're, piddling in the ocean, if you will. And yeah, you're not going to, really the risk of benefit is less. Pediatrics, we've argued successfully that we often start at about 80% or close to the adult dose. So you're right that you start at a level where you don't think you're going to have side effects, and that's the goal. And we do everything, and I'll tell you, I've had studies stop when some wacky side effect comes out until we can figure out what's going on. So we do our very best. Nobody wants any horrible side effects. But the pediatric dose, we do start a little higher because I don't want to waste these valuable resources of patients and giving them a dose of a drug that's not going to work. So I often argue for a slightly higher dose than the piddly dose that the adults start with.
Any other questions? No? Okay. Thank you, Dr. Macy.